Today we are going to be discussing prokaryotic catabolism, or ATP synthesis. ATP synthesis in prokaryotes can be achieved in a number of ways. We are only going to focus on 3 of them. Aerobic cellular respiration, which requires the use of oxygen, anaerobic cellular respiration, and fermentation, both of which do not use oxygen. Before we can understand how prokaryotes produce ATP, we need to do a review of a particular type of chemical reaction called the redox reaction. It's actually 2 chemical reactions that are paired together. Whenever there's one of these reactions, there has to be the other reaction as well. In a redox reaction, one molecule loses an electron and another molecule gains that electron. So basically this is the transfer of an electron from an electron donor to an electron acceptor. Cellular respiration, whether it be aerobic or anaerobic cellular respiration, is the complete oxidation of glucose. What that means in plain English is that during respiration, all of the electrons are removed from the glucose molecules. These electrons are removed from glucose and then they're given temporarily to electron carriers, specifically NAD+, which then becomes NADH+, plus H+, which biologists will then shorthand to NADH2. And they're also given to FAD+, which becomes FADH2. So those electrons are donated to these electron carriers, and the electron carriers will ultimately take them to the electron transport chain, or the ETS, where those electrons are used to catalyze ATP synthesis. Sometimes respiration is called the NAD cycle, because all it really is a series of redox reactions where we remove electrons from glucose in a stepwise fashion, give them to the electron carriers, and then the electron carriers take them to the electron transport system. So cellular respiration occurs in a stepwise fashion, and we break it down into 4 subpathways so that it is easier to understand. The first of which is glycolysis, which is the initial breakdown of glucose. The transition reaction, which connects glycolysis to the Krebs cycle, also known as the TCA cycle, and ultimately ending with the electron transport system, also sometimes known as the electron transport chain. Those terms are used interchangeably. We are going to go through each one of these subpathways in order and in some detail. Starting with glycolysis. Glycolysis is defined as the breakdown of 1 molecule of glucose into 2 molecules of pyruvate. So we start with 1 glucose, and it's broken down in a stepwise fashion into 2 molecules of pyruvate or pyruvic acid. In order to get this reaction started, you can see that we spend 2 ATP. So right at the beginning, we spend 2 ATP molecules. You know, you have to spend money to make money. You have to spend energy to make energy. And this part of glycolysis then is known as the energy input stage of glycolysis. What this ATP does in the beginning of this reaction is it destabilizes the glucose molecule so that it can be broken in half. Glucose is actually a 6-carbon molecule. So if I just draw it, a rough drawing of glucose here, 6 carbons, and what we've done to the glucose between here And here is we've added this giant negatively charged phosphate on this end and another giant negatively charged phosphate on this end. These two phosphate molecules want to get away from each other, so they repel each other, and this causes the glucose molecule to split in half down the middle. And that is going to give us the lysis stage right here where the glucose molecule splits. Now, because we split the molecule in two, everything that happens from here on out happens twice. And I've shown it here with both sides, but in some renditions we only show this happening once, and you have to multiply all of these chemical reactions by 2 because they are happening twice. So as we move along in continuing the breakdown of our glucose molecule, here you can see we're starting to pull off some of those electrons and give them to NAD+ to make NADH2. So right here we have made 2 NADH2 molecules. We've used 2 ATP. As we continue the breakdown of our glucose, we make 2 ATPs. So we're up 2 ATPs, and we continue down through the conversion and the breakdown of glucose into pyruvate, and we make 2 more ATPs. So we've made a total of 4 ATP, but because we used 2, this is a minus sign here, our net gain of ATP is only 2, and we've also made 2 NADH2s. Now, we are trying to make ATP, but it's important to remember that the bulk of our ATP production ultimately is going to come because of these NADH2s. So as you're following along in your notes, you're filling out your notes page, 1 molecule of glucose is broken down into 2 molecules of pyruvate. We make 2 ATPs and 2 NADH2s. Now those 2 pyruvic acid, so I'm only showing 1 here. Remember, there are 2 pyruvate or 2 pyruvic acid in the transition reaction are going to be broken down into 2 molecules of acetyl-CoA. So this part of it is the transition reaction right here that I boxed in, because the transition reaction is what connects the glycolysis to the Krebs cycle. So 2 pyruvate are broken down into 2 acetyl-CoA. So we get from that 2 more NADH2 molecules. So I've only shown it once, but I'm multiplying everything by 2 in my head. Carbon dioxide is given off as a byproduct of this reaction. And now this acetyl-CoA is free to enter the Krebs cycle, which we're going to see in the next slide. So keeping track of our chart at the beginning of, or at the bottom of your notes page, we've made 2 ATPs during glycolysis, 2 NADH2s during glycolysis. We don't make any ATP in the transition reaction, but we do make 2 NADH2s. So that Krebs cycle, our acetyl-CoA is going to enter the Krebs cycle, and it's a circle. So I'm not going to ask you what it begins with and what it ends with. That really depends on your point of view. I hold with the idea that these 2 carbon molecules from acetyl-CoA join with these 4 carbons from oxaloacetate to make the 6-carbon citrate molecule. Other people say that the Krebs cycle begins with the citrate molecule. It really depends on your point of view. And I don't want to get into that sort of hair splitting because I really want to stick to the big picture, which is the number of electron carriers we generate and the number of ATP molecules that we generate. I'm not going to make you memorize this Krebs cycle. You do have to remember that the Krebs cycle is turning 2 times for every glucose that enters cellular respiration. So all of this stuff is happening twice. So as the acetyl-CoA enters, it combines with the oxaloacetate to become citrate. The citrate is converted into an intermediate, which is converted into another intermediate. I'm not making you guys name these. We are going to generate NADH2, which, remember, there's going to be 2 of them. We got another intermediate. We make some more NADH2s, 2 more. And as we continue around the circle, we make GTP, which is then very quickly converted into ATP. And again, we're going to get 2 of them. Continuing around the circle, we make some FADH2s and 2 NADH2s at the end, at which point our oxaloacetate is regenerated so that the Krebs cycle can continue to turn to generate the electron carriers and the ATP that the cell is going to use for energy. So double checking back to our chart, we've made 2 ATP during glycolysis, 2 NADH2s during glycolysis, and no FADH2s. In the transition reaction, all we have made is NADH2, and in the Krebs cycle, we have made 2 ATP, 6 NADH2, and 2 FADH2. Now, so far, all of the ATP that we've made in glycolysis and the Krebs cycle, these were made by something called substrate level phosphorylation. And all this means is we've made the ATP without the benefit of the electron transport system. And so far, we've only made 4 ATP. In prokaryotes, per glucose molecule, we should be getting 38 ATP. We've only got 4. So the bulk of our ATP are going to come from the electron transport system. So within this electron transport system, it is a series of enzymes embedded in the cell membrane of our bacterial cell. So you have to remember, bacteria don't have organelles, so they don't have mitochondria. This happens right in their cell membrane. So on this side, this is the inside of our bacterial cell. This is the outside of our bacterial cell. Each one of these blue structures is an enzyme, and these little things are enzymes as well. So our NADH2, our FADH, they are going to come to the electron transport system, and you can see here's our NADH2. It's going to drop off this electron to the enzyme. Now this electron is going to be passed from this enzyme to this enzyme to this enzyme to this enzyme to this enzyme, eventually until it bounces back into the cell. The electrons are activating basically what amount to proton pumps. The electrons trigger these to pump hydrogen ions outside of the cell. Now here's the big picture here. This is, if you guys don't get anything else from this is the big idea, the big picture. This is how all the ATP is generated, is this accumulation of hydrogen ions. When we have a situation where there's a lot of something on one side of a semi-permeable membrane, but not much of it on the other side, we know that that is a gradient. And cells are not particularly fond of situations where things are out of balance. So they're always striving for equilibrium. So what the cell wants to happen is for these hydrogen ions to come back in and establish equilibrium. Now the problem with that is these pumps are one-way tickets. You can only go out. The hydrogen ions cannot come back in through these pumps. Also, because of the hydrophobic nature of the cell membrane, hydrogen ions can't pass freely through the membrane. So now we have this situation where we have a gradient. The cell is striving for equilibrium. These hydrogens want to come back in, and the cell has devised an elegant solution to this plan. This is my favorite enzyme. If you can, I'm such a nerd. I have a favorite enzyme. This is my favorite enzyme. It's called ATP synthase. So you can tell by its name synthase, it's synthesizing ATP. And the way this enzyme works, it's a very, very cool enzyme. It allows the hydrogen ions to flow back into the cell. But they don't just travel through this thing. It's not like a tunnel. It's not shown in this diagram, but there are actually moving parts inside this enzyme. And so the flow of hydrogen ions through it causes the moving parts of the enzyme to turn or to spin. So basically we are converting chemical energy into energy of motion. This is not unlike what we see with a wind turbine. The wind causes the wind turbine to spin. That is energy which is then captured into batteries. So in that case, we're converting energy of motion into chemical energy or stored energy in a battery. The energy generated by the flow of these hydrogen ions through ATP synthase is enough to catalyze the reaction ADP, or adenosine diphosphate, plus phosphate into ATP. It takes a lot of energy to do this because ADP already has 2 phosphate molecules on it. They're negatively charged. Phosphate is negatively charged. They don't want to go together naturally. It takes a lot of energy to catalyze this chemical reaction, and they achieve that by creating this energy of motion to get those molecules to go together and create ultimately our ATP molecules. The other part of this reaction that we need to deal with is the fact that our electrons then come back bouncing in here, and we can't just allow these electrons to be free in the cell and bounce around, because when they do that they create damage. So the electrons have to be picked up, and they're picked up by something called the final electron acceptor. In this case, we're talking about aerobic cellular respiration. So the final electron acceptor is oxygen. That's what makes it aerobic. Oxygen is the final electron acceptor. So the electrons come in, they combine with the oxygen and some of the hydrogen, and that gives us, as a byproduct of aerobic cellular respiration, water molecules. The final important piece to this situation is the conversion of NAD plus or NADH2 back to NAD plus. Okay, so as our NADH2s donate the electrons to the electron transport system, they are automatically converted back to NAD plus. That means that this reaction can continue to occur again and again. The cell is going to continuously need to make energy to survive. So these NAD pluses go back to the cytoplasm where they pick up those electrons from glycolysis and then bring them back to the electron transport chain in the form of NADH2 again. So calculating the total energy production from 1 molecule of glucose. So we always are doing this in relation to 1 molecule of glucose. For every NADH2 that is produced, 3 ATP are made. So if we look at our chart over here, we made 2 NADH2s during glycolysis, 2 during the transition reaction, 6 during the Krebs cycle. That gives us a total of 10 NADH2s. So if I multiply 10 NADH2 by 3 ATP per glucose, that's going to give me 30 ATP molecules from the NADH. Then if I look at my FADH, 2 ATP are made per FADH2 molecule. I only made 2 FADH during this procedure, so 2 FADH2 times 2 ATP per glucose gives me 4 ATP from the FADH2. So if I start adding this up, now from the electron transport system, I get 30 plus 4 gives me 34 ATP. And then I go back and I look, I made 2 ATP during glycolysis and 2 during the Krebs cycle. 2 plus 2 is 4. So I get 38 ATP total during respiration. And so that's where all of the different numbers come into play. So 38 ATP molecules total during respiration, 4 of them from substrate level phosphorylation, the remaining 30 from oxidative phosphorylation from the electron transport system. So anaerobic cellular respiration. Now that we've been through aerobic cellular respiration, we need to cover anaerobic cellular respiration. Now this is about the time most people start to freak out because this is a lot of molecular biology, but it uses the 4 same subpathways as aerobic cellular respiration. The difference is oxygen is not the final electron acceptor at the end. And to simplify things, we're going to say that the final electron acceptor is anything but oxygen. So bacteria are very, very diverse in how they can make ATP. So we're not going to get into the nitty-gritty details of this because we could spend an entire semester on bacterial metabolism. So we'll just simplify it and say it's anything but oxygen. For example, if they use sulfur as their final electron acceptor instead of water, they give off sulfide as the byproduct of their metabolism. If they use carbon dioxide as their final electron acceptor, the byproduct is methane instead of water. So they can use lots of different things like nitrogen, all kinds of things that we're not going to go into much detail on. But it's the same 4 subpathways. They still do glycolysis, the transition reaction, the Krebs cycle, and the electron transport system. So here we have the electron transport system. This is anaerobic cellular respiration, and we can see right here 2 hydrogens. The final electron acceptor is not oxygen, it's anything but, and so we get some byproduct other than water. And our final reaction for making ATP is fermentation. Fermentation is an anaerobic process. It does not use oxygen. However, it can occur in the presence or absence of oxygen. It's much less efficient than respiration because it yields only 2 ATP per glucose instead of 38. It's defined as being essentially glycolysis plus an additional step. And that additional step is used to oxidize the NADH2 back to NAD+ so that energy production can continue. So remember I told you at the end of our discussion on the electron transport system that this conversion happens automatically when the NADH2 donate their electrons to the chain. We automatically get this NAD+. But because fermentation is glycolysis, this conversion from NADH2 back to NAD+ does not happen, and so they have constructed an additional step to ensure that that occurs. And another fun thing about fermentation is this additional step creates byproducts, many of which are useful to humans. So this additional step varies from bacterium to bacterium. It just depends on what enzymes and organic molecules are present in the bacterial cell. And so some of those bacteria or yeast will make alcohol as their additional step. They might make isopropanol, they might make butane. There is a huge range of things that they can make during fermentation that humans find useful or delicious, like lactic acid. Fermentation of milk gives us cheese and yogurt. Obviously, alcoholic fermentation gives us alcoholic beverages, and so on and so forth. So with fermentation, it is just glycolysis. So we start with 1 glucose molecule. it's broken down into 2 molecules of pyruvate. And then here we can see the various additional steps that are possible. And this is only a few of them. So our pyruvate can be converted into lactate or lactic acid. As I mentioned, that's going to give us milk or cheese or yogurt. If the additional step gives us propionate, that gives us Swiss cheese. It's a little bit different. Acetate, ethanol. So there you got your beer, your wine, your liquor. You make bread during alcoholic fermentation. Butanol, so this would be lighter fluid. Propanol, acetone. So these are solvents that humans find useful. So it just depends on what type of microbe we're working with. This additional step is going to be different. And that is going to convert our pyruvate into this useful byproduct and regenerate our NADH back to NAD+, so that the cell can continue to produce ATP. So again, these fermentation end products are very useful to humans, not just because we like to eat them or use them to remove our nail polish or use them as antiseptics, but they're also important in bacterial identification. So we get useful products, but also we can identify bacteria based on the byproducts they produced during fermentation. So if a bacterium produces lactic acid, we can look backwards and say, well, the genera Streptococcus, Lactobacillus, and Bacillus make lactic acid, so maybe our unknown bacterium is one of those 3. If our unknown bacterium makes butyric acid, butanol, acetone, isopropyl alcohol, and carbon dioxide, we can look back and say, well, the only genus I know of that makes all of those things is the genus Clostridium. So maybe our organism is a member of that genus. So these end products are very important in identifying unknown bacteria, because the bacteria have these metabolic profiles where they each produce different byproducts during fermentation. So the relationship between metabolism and growth, I always cover these topics together because metabolism is essential for growth, and growth is a metabolic process. So obligate aerobes are bacteria that require oxygen, therefore they undergo aerobic cellular respiration. Obligate anaerobes are a type of bacterium for whom oxygen is toxic, and so they make ATP via anaerobic respiration or fermentation. And finally, facultative anaerobes can switch between respiration and fermentation depending on oxygen availability. So it depends again on the type of bacterium. Some are very rigid in their requirements, and others have options, and they can switch back and forth. So if we take our obligate aerobes, we put them in an environment with no oxygen, they're going to die. And the same thing with our obligate anaerobes. If we put them in an environment with oxygen, they're going to die. Our facultative anaerobes don't care. Our facultative anaerobes would prefer to respire because they make 38 ATP during respiration, but if you take them out of that environment and they don't have oxygen available, rather than die, they'll switch to fermentation and they can live off 2 ATP per glucose molecule. So this is an advantage to them for their survival, is that they can switch their metabolism back and forth depending on the environment that they're living in. So I really want to get to this big picture about metabolism because it's such a molecular topic and a lot of people don't enjoy the chemistry behind it. So I really want to share with you why understanding metabolism is so important for biologists, especially microbiologists. If we understand the metabolic pathways of the bacterial cells, we can develop ways to disrupt that metabolism. And we also need to be aware of what metabolic pathways they have in common with us. So if their metabolic pathways are similar to ours, it's harder to disrupt their metabolism. And it's also important to know if we're dealing with an obligate aerobe, an obligate anaerobe, or a facultative anaerobe. So let's just pretend for a minute that I'm dealing with an obligate aerobe. So it not only needs oxygen, the obligate aerobe needs oxygen, but it also needs to make ATP by starting with glucose, completing glycolysis, completing the transition reaction into the Krebs cycle, completing the Krebs cycle, and using the electron transport system to make those 38 ATP. It doesn't have an option. It can't switch. So with our obligate aerobe, if we remove oxygen, there's no oxygen here to be the final electron acceptor, that means that this final step isn't going to occur in the electron transport system. So all of these reactants are going to get backed up here, and eventually the electron transport system stops, the Krebs cycle stops, glycolysis stops, and this cell is not going to have any ATP. So no ATP, and this cell is going to die. So we're going to have a dead bacterial cell. Now, if we back this up a little bit and say, well, what if it's not, what if there's oxygen present? What if we fiddle with this at another point in the pathway? Maybe we disrupt this enzyme here in the Krebs cycle. Well, that's going to cause the Krebs cycle to stop. We're not even going to move into the electron transport system. And again, we're going to end up with a dead bacterial cell because it can't complete metabolism. But what happens if we're talking about a facultative anaerobe? If we go back to the first example and say, oh, hey, look, let's take away the oxygen here and disrupt this step. Well, that means the electron transport system is going to stop. But a facultative anaerobe can switch its metabolism out of respiration and just do fermentation. So it will back up and it will just do glycolysis, and it's going to switch to making 2 ATP per glucose versus those 38 ATP from the electron transport system. So it'll survive because it has that option. If we were, say, maybe to disrupt the Krebs cycle or the transition reaction, let's just take out this enzyme. Now the Krebs cycle stops, which means the electron transport system stops, but glycolysis is still intact. So it's just going to go back here and it's going to do glycolysis and it's going to continue to survive. But what happens if we destroy one of these enzymes? Any of them. We can that one or that one. All of these arrows are enzymes. If we destroy any of these enzymes within glycolysis, then glycolysis is done too. And so now even the facultative anaerobe is in trouble, and it will die too. So the facultative anaerobe does have a bit of an advantage for survival in that if we destroy the Krebs cycle, if we destroy the electron transport system, it can switch over to glycolysis. But if we mess with glycolysis, we can kill the facultative anaerobe too. So it's important to understand metabolism and which points in metabolism you can mess with in order to kill these bacteria and not interrupt your own metabolic pathways. So for your next steps, and as you're answering the questions on your assignments, make sure that you know the amount of ATP, NADH2, and FADH2 produced during each subpathway of respiration. Make sure you can describe how the hydrogen ion is gradient is generated in the electron transport system and describe the importance of this gradient. Make sure that you can describe how ATP synthase works to produce the ATP. Make sure you can describe the process of fermentation, the importance of those fermentation end products, and make sure you can discuss what happens to a bacterial cell when metabolic pathways are disrupted. I will not ask you to memorize the steps of glycolysis, the transition reaction, or the Krebs cycle. The things listed here are the things that I expect you to know for this chapter, for your quiz, your written assignments, and your exam.